Hello, Evelyn.
No.
How are you doing?
All right. How about you?
Not bad, not bad. All right. All right. So I'm going to let's see what we have prepared for this week. So here are the tasks that are prepared for this week. We're going to go over chapter 12 and chapter 13 regarding neural tissue. Some of these chapters regarding the nervous system, they're going to be just thick in root memorization, the name of nerves, the name of regions within your brain. We are complex creatures. We have a complex uh, neural architecture in general. And in order for us to learn that, we just have to repeatedly expose ourselves to the different nuclei, the different centers, the different regions. We're going to divide it and structure it in a way that it's the most comprehensive. However, this is a complex topic, and thankfully it is a complex topic, because if it wasn't, we'll be considerably simpler than we are. So let's get started. We have these following two tasks that are going to be that are opportunities for bonus. All right, so let's go to chapter 12, the nervous tissue. I know that some of you folks have had issues with either an exam or a quiz. I've mentioned the protocol to deal with those issues is please join these meetings, and then at the end of the meeting, you let me know what the specific issue is. We'll handle it, and we can move on. I hope that so far I have several emails about issues that have occurred either during a quiz or during an exam. But so far, I haven't had one person request that we talk about those issues in person. So I'm afraid that at the end of the semester, I'm going to have a lot of requests for that. So this is the time to get them over with. I'm going to put a hard deadline on them. Any requests that are so we'll divide them in two sections before exam 2. We'll handle all of requests that occur before that deadline, and we'll handle the requests after exam 2. But you can't come to me at the during our last class and tell me that you have issues with exam 1. That's just, this is the time to do it. Let's try and do it. It's we can do this. Let's just do it. Okay. So, and that it's probably for folks watching at home. All right, let's get this started. Chapter 12. Neural tissue. Can you see my PowerPoint? Excellent, thank you. All right, let's go with neural tissue. What are our objective is describe the major divisions of the nervous system. As you may imagine, there's a functional division of the nervous system, and there's some structural divisions of the nervous system. Structural divisions, you can consider that the central and the peripheral nervous system. Functional divisions of the nervous system includes the nervous system that you can control and the nervous system that is beyond your control. For example, I'm digesting at the moment, and I don't have to consciously think about that. I don't have to consciously make a decision towards digestion. I'm breathing, and I don't have to constantly think about every breath I take. My heart is beating. That in itself, we mentioned there are pacemaker cells within the heart, specialized cells that take care of this. However, there is your heart rate is also under control of the parasympathetic nervous, of the autonomic nervous system, the involuntary nervous system, and you don't have to think about whenever you need your heart to beat faster. Whenever you're running, you don't make a conscious decision for your heart to start beating faster to pump more oxygen into muscles that are needed. So those are functional divisions of them. Structures of a neuron, neuroglia, resting potential, action potential. Let's do all of that. The synapse, neurotransmitters, neuromodulators, process information with neural tissue, all of that fun stuff. Nervous system, okay, it's here. So what I want you to, the reason why they're bringing the endocrine system and the nervous system here is because both systems are a way for us to react to the environment. So usually you can think about the endocrine system. We talked about it in terms of homeostatic levels of a particular homeostasis that we maintain, and then any deviation on either direction will be counterbalanced will have a response by the endocrine system, by hormones. So those deviations from homeostasis, those are responses towards not only those particular homeostatic levels, but also our environment, responses towards consumption of food, responses to changes in temperature, responses to our I don't know, dehydration and so forth. But the nervous system is the endocrine system. It's a little harder to think about it as solely as our responses towards the environment. That's why, but it's easier to think about the deviations from this homeostasis. However, if we, I think we can do mental gymnastics to get it to fit that definition. The nervous system is more intuitive in terms of responses to our environment, responses to stimuli. The one thing that in this case, I hope that it's the one clear distinction between the two is that the nervous system, it travels at the speed of an action potential, which is pretty fast. Whereas the endocrine system, it's a considerably slower process. It requires the release of the production of hormones, the release of hormones into vesicles. Where are the hormones being released? So let's say your thyroid hormone, your insulin. your kidney is making insulin. Where does it release it? So a cell, I'm a beta cell in the pancreas. Sorry, I said kidney. In the pancreas, I'm a beta cell in the pancreas. I sense that you had a large cupcake, and in response to that, I make insulin so that that sugar that we consume, we send it into cells. So I make that insulin. I make it in vesicles. Now, where do those vesicles go? Where do vesicles go in general? I wanna start at the very detailed view. A cell makes a vessel. That's how cells communicate with each other. They make stuff. They make proteins. They put it in vesicles, and then they exocytose those vesicles, right? Just like I think it's more intuitive in whenever we talk about neurotransmitters, right? We have dopaminergic neurons. We have cholinergic neurons making acetylcholine, making dopamine. Neurons making serotonin. They put them in vesicles, and then those vesicles get exocytosed. Same goes for beta cells in your pancreas making insulin. They make insulin, put it in vesicles, and they get exocytosed, release. Now we know where the neurotransmitters go. They go into a synapse, right? Now where does the insulin, from these vesicles go? So it goes to the outside, right? It goes to the outside. In the case of a neuron, it goes to the outside. In both cases, it goes to the outside. In cases of a neuron, they release this specialized the specialized exterior of the cell called a synapse. That's just a specialized place that where two neurons communicate with each other. In terms of most hormones, they're released to the exterior environment. Usually, we talked about how no cell is farther away than 100 micrometers from a blood vessel. So whenever they release these substances to the exterior environment, they're fairly close to a blood vessel. And that's where that's how your pituitary gland is able to make a hormone that gets just by diffusion it diffuses and enters a blood vessel, and then it goes to every cell of your body, and that's why you can have luteinizing hormone released from your pituitary having effects in your ovaries because it was made in a vesicle, got released into the exterior environment. There's a blood vessel everywhere, basically, and so the hormone diffused into the blood vessel, and then the circulation carrier everywhere. It only affected the luteinizing hormone only affected follicles, maturing follicles within ovaries, because they were the only ones that had the receptor for it. All of the other cells, they don't recognize it. It's kind of like a lock and key mechanism. It just flies by them, no receptor. But whenever a cell has a receptor to a hormone, a reaction occurs, and then we see the downstream effects. Point being, it takes longer than what an action potential through the nervous system does. So you can imagine your nervous system. kind of makes sense that it will be the one that controls your these behaviors that need to be immediate. For example, movement needs to be immediate. If you did control movement to hormones, then by the time all of that happens, the vesicular release, the circulation throughout the blood, the lock and key receptor, the downstream effects of it, it would take a while before we can react to an environment. And usually environments, at least in the wild, wherever all of these systems evolved, used to be considerably more dangerous than it is now, and where we needed to pivot immediately. But anyway, divisions of the nervous system: the central, the peripheral, and within the peripheral we have neurons that are sending a signal outside, away from the central nervous system, and neurons are sending a signal towards the central nervous system. So let's look at this major division. This is the central nervous system, your brain and your spinal cord. That's easy enough. And then you have the peripheral nervous system, which includes all of these nerves. Now, these nerves, we mentioned that the nervous system is involved in these immediate reactions. One of them is sensation, right? So whenever I run my finger through the palm of my hand, I feel there's a sensation that comes. So a signal we must be activating a signal that takes a sensation from here towards the spinal cord, towards the central nervous system, and eventually towards my brain cortex, that corrugated area that we know as brain, the corrugated area on the surface of the brain. So that is happening. There's a signal being sent here. Now, let's say that I push harder and I want to, I have a reaction, an aversion. So there was a signal that was inputted, but there's also an output signal that made me flinch or react. That an output signal that told my nerves do something. And that output signal, once it starts once again at your neocortex, at your brain cortex, and that sends signals down the spinal cord, and eventually out of the spinal cord through these peripheral nerves and into the muscles within my hand that allow me to react. Point being, signals can go in and signals will go out. If a signal goes into the central nervous system, it's called an afferent neuron or an afferent signal. Efferent, same as exit, it goes away from the central nervous system. Now, signals that go into the central nervous system are usually sensory signals, right? Signals that tell you, hey, you're feeling this, or whenever you overstretch a muscle, it's telling you, hey, calm down, don't overstretch that muscle. Even though that signal is coming from the muscle, it's still a sensory neuron that's sending it, the signal. And so the signals that are going in are sensory. Signals coming out, we are reacting in a way to whatever stimuli to this type of stimuli, and those output signals, the signals are coming out, usually involve either glands or muscles or something that reacts to this sensory signal. So let's start from this end. The sensory so here are we have receptors that allow us to sense these signals. We have different types of receptors. We have special sensory receptors. So that's your typical what you think about your special senses, your vision, smell. But whenever if I were to ask you about receptors, you probably answer those. Now let's look at specialized types of these receptors. We have visceral sensory receptors. So these are receptors that are monitoring, that are also, there's these signals that we're monitoring, or these sensory signals that usually for the external environment. We also have sensory input from inside of us, right? Sensory input that tells me, oh, your stomach, it's a little distended, you ate too much. Or sensory input that allows me to, so whenever you have to go to the restroom, you have this sensory input sending that signal into your brain. Whenever you are your heart rate, your respiratory rate, all of those kind of visceral organs are being monitored. All of these systems, they're being monitored and sending a signal, letting your brain know, or letting the organism know that your respiratory system is functioning properly, your cardiovascular system is doing what it should be doing, your digestive system, stop eating, it's distended, you ate too much. Your urinary system telling you that you need to go to the restroom, or not only that, but also once we get into all of the functions of the urinary system, whenever it comes to regulation, homeostasis of ions, all of those signals are going to play out a role here. So now we looked at the receptors for the external environment. We looked at receptors for these internal organs, internal systems. Then also we have receptors that tell us where we are in space. So they will monitor your joints. They will tell you, for example, your hand is up here. You need to know that your hand is elevated. And there are receptors that will inform you that your hand is elevated. That way you can act from there. Or that there is that you are hyperextending, that your tendons are hyperextending. So you will be able to, we need to sense that. Okay. And I see vision hearing. These somatic receptors will also include your integumentary system. So that's the special sensory are basically anything that's in your head area, okay? And the somatic receptors, they are your integumentary system, your skeletal and muscular system. Okay. So those are basically all of the systems, all of the sensory receptors that give us information about ourselves, about our environment. Now, those signals we mentioned, they're going into the central nervous system. So what's the name of that, of those neurons, of that division? It's called afferent. So these nerves are sending signals into the central nervous system. Since they're going into the central nervous system, they're called afferent. Now, it goes from the peripheral to the central nervous system, higher brain order centers. They communicate several systems within your cerebral cortex talk to each other, and they integrate information because Most systems, they are constantly interchanging information, and your response is going to depend on the interaction of these thousands and thousands of millions of inputs from all of these systems simultaneously integrating, and then we coming out from the efferent division of the central nervous system. So we integrated all of that. Now we respond. There is these neurons are formulated. This is what we're going to do. And now it goes from the central nervous system to the peripheral nervous system, the efferent division. We have, whenever it comes to our response to it, it can be voluntary, right? I'm going to move my hand in response to a poke or whatever. And there are involuntary. We had all of these sensory mechanisms that were looking into our heart rate, or looking at our respiratory system, or looking at our urinary system, or looking at our position within space. Now we formulated our response. One of them, the voluntary one, is kind of intuitive. The skeletal system has been involved. The involuntary, and that's the somatic nervous system, the voluntary one, and the autonomic nervous system, the ones that it's beyond our control. One thing that I want you to pay attention is the concept of somatic. That means somatic, the body. Whenever we talk about the body's sensory receptors, this is your body. It's talking about mainly your skeletal, osseous, and integumentary system, considered as body. And in this case as well, the somatic nervous system. So it's your body's nervous system, just like skeletal, osseous, and integumentary system. Anyway, the response goes to skeletal muscles, and we have a response. The autonomic one, it's not under our control. The autonomic one works in two functions. The one that activates, the one that kind of goes in one direction, and the other one that goes in the opposite direction. So one that increases your heart rate and one that decreases your heart rate. One that increases your respiratory rate, one that decreases your respiratory rate, one that increases urine production, one that decreases urine production, increases digestion, decreases digestion, increases the function of the immune system, decreases the function of the immune system. All of these things all of the things that I mentioned, you don't control, you don't make a choice to do them. The autonomic nervous system is doing, and it's doing it in this system that is opposing to each other. For the most part, you think about you have the sympathetic and the parasympathetic. The sympathetic, that's your fight or flight. That's where you probably heard of the sympathetic nervous system more often, your fight or flight. So there is something that triggers the sympathetic nervous system. Sympathetic nervous system activates itself, and it leads into this series of events that are conducive towards sudden energy expenditure. So you know, all of a sudden, a tiger shows up in your room. Everything that you need to do to escape that tiger, that's what your body is going to do. So what do you need to do to escape that tiger? You need your pupils to dilate for hyper focus, to get more lighting. This is the moment. You're sending more energy towards your senses, towards your vision, in order for you to react appropriately. Energy is being deployed into your skeletal muscles in order for you to have this like ridiculous amount sudden burst of strength that you can react and fight this target away. Your heart rate is increasing because you're going to need a lot of oxygen for the acrobatics that you're going to do to escape this tiger. All of these events that you can think of, respiratory rate is increasing, heart rate is increasing, all of these things that you do in order to fight this, your sympathetic division of the nervous system are performing that. Now, jokes on you, like there's no tiger in there. It was just like, I don't know, you just drank some bad milk or something. So the sympathetic division is no longer, the parasympathetic division of the nervous system takes hold. And basically the parasympathetic division of the nervous system, is, like we mentioned, there's some things that increase your heart rate, there's one that goes the other way. Parasympathetic division is like business as usual. Let's go back to homeostasis. And that's how you should think it, even though the one increases, the other decreases, the sympathetic is more like, let's go back to homeostasis. All right. So did we finish that? Okay, so the parasympathetic, sympathetic, they act upon, this is the effector, so it makes sense that for the somatic portion, the one that you're going to control is skeletal muscle. You control skeletal muscles. You don't control cardiac or smooth muscles. Those are beyond your control. The autonomic nervous system, the one that is modulating this, that is modulating these behaviors, these reactions. That one is under the control of smooth muscle and cardiac muscle. Okay. Now let's look at the workhorse of the functional unit of the nervous system. This would be a neuron. Here you have specialized nomenclature for a neuron. You have your pericardium, and this is part of the cell. So here you have so you have your pericardium, which whenever it comes to the neuron, these are all specialized names of structures that we already know. So that's the joy of anatomy of basically science in general, that anatomy and physiology, that depending on where you are, these particular cells or these structures, even structures within their cell, will take on specialized names. So here you have the pericardium, which is basically the cytoskeleton of the neuron. It contains neurofilaments, which are the neurofilaments, neurotubules, neurofibrils. So this is the analogous intermediate filaments, analogous to microtubules, analogous to actin fibers. That's all they are, but they are for the neuron. Axon here is, let's just go into the animation, the photo itself. So this is what a cell looks like without, if you take away the axon, you have your stereotypical cell here. Except for the dendrites. Usually neurons do not have dendrites, or other cells do not have dendrites. But neurons do, but in terms of the organelles, you have your nucleus, you have your endoplasmic reticulum, you have your mitochondria, just good old organelles. And then neurons, we have this, the cytoskeleton that we mentioned. That's the one that gives neurons these specialized structures. These are filled with the cytoskeletal materials. Uh, Neurons are nothing than or cells in general. They are basically a lipid bilayer with water in the middle. Now, I think you have an intuition of the texture of a lipid bilayer. It's like a layer of oil on top of water. There's not much structure to that. So where do they get this complex structure? That's where the cytoskeleton comes into play. The cytoskeleton itself gives it this complex structure. These complex structures allow neurons to form dendrites. So this is all of these microtubules from the neurotubules coming here, and then you have this, or most of this is going to be intermediate filaments and microtubules as part of the cytoskeleton. And then at the tips of each one of these, you have an actin filaments. So actin filaments basically at the membrane level is kind of like coated with actin filaments. And that particular disposition is what provides allows this very viscous type of lipid bilayer to create this kind of more complex structures. All right, so here we have, so these are dendritic spines. So you have your dendrites, and within the dendrites, there are specialized little knobs or bulbs that are called dendritic spines. Their name kind of is fairly descriptive. They look like spines coming out of the dendrite, out of this branch. And these spines is where synapses are occurring between different cells. Synapses are occurring at all levels of this neuron. But for the most part, they're concentrated within these spines. And you have here your axon helix. Your axon helix is we're going to look at an action potential, and then we'll kind of understand the relevance of the axon helix in terms of this is where the action potential kind of generates. This is where the cell decides, hey, am I going to fire an action potential or not? What gives? Once it fires that, it goes down the axon, and then from the axon, we talked about there are vesicles at the ends of this axon, these axon endings. There are vesicles at the ends of these synaptic terminals. And those vesicles, when the signal arrives at the end of the neuron, that signal tells those vesicles fuse with the membrane. So those vesicles that were living inside of these synaptic bulbs, they fuse with the membrane and they release their components. That awesome analogy that we were talking about, that how things fuse. Anyway, that's how we release neurotransmitters. And we will go into the detail of how a cell that gets thousands upon thousands of inputs, how does it decide whether or not to fire an action potential, right? That's what's happening here. It's like thousands of other neurons are sending a signal telling it. So you probably have a neuron coming from your sensory system. You have a neuron rising from, as I speak to you right now, you're probably indulging neurons from your sensory system, because you always are. Your visual system, your auditory system. You integrate those signals reach your cerebral cortex, and those signals go to integration areas, second order integration areas, where those signals you kind of provide some context into it, then they communicate with each other. Eventually, you get to a place where you're kind of forming an idea of what this is, of what's occurring. You're putting all of this together, visual, auditory, sensory signals, everything. And that's only including, we're only talking about the the sensory division. We're not talking about yet about somatic or visceral division of these senses, but all of those are occurring, and you kind of put it all together in your front. There's a lot of integration occurring at these higher areas, and then in your frontal cortex, you're kind of having this idea, this intuition of what is happening. So all of these signals are integrating at all of these levels, and eventually one neuron, a population of neurons is getting all of the signals from all of these places, or signals that have gone through all of these integrations at other levels, and whether a neuron will fire or not based on all of these inputs that is coming to it. Some of them are going to be excitatory, some of them are going to be inhibitory, and it kind of adds up all of those inputs, and eventually, if it comes down to whether all of those signals reach a threshold for firing action potential, whether this neuron is going to react or not. And the currency of the neuron is an action potential. So it's either yes or not. Am I going to fire one or not? This occurs whether if there is a firing, the communication goes into the neurotransmitter is released, and it goes into the synapse. We mentioned you will hear the talk of the presynaptic and the postsynaptic neuron, right? The neuron that sends the signal and the neuron that receives the signals. That's all there is. The neuron that makes the neurotransmitter and the neuron that receives the neurotransmitters. The neuron that makes dopamine, the neuron that has the dopamine receptors to respond to that dopamine. These neurons are separated by the synapse. What do we mean? We mentioned the synapse was the outside environment, right? That's all. Whenever we release a vesicle, it's released into the outside environment. The synapse is kind of like a specialized outside environment. It's a very controlled outside environment. And in this case, here you have your what's called the synaptic cleft. This kind of this portion that divides the two neurons, basically. At the dawn of neuroscience, there was a debate between these two neuroscientists, Antonio Ramon y Cajal, and that's probably where your stereotypical neuron diagrams and neuron photos, they were done by him. And I can't remember his name, her first name, but his last name was Golgi. And if it should sound familiar because you heard of a Golgi stain. But these two scientists at the dawn of neuroscience, they didn't know whether the neurons were individual cells or whether it was just one whole syncytium, one whole meshwork that shared the same cytoplasm. They didn't know that. As microscopy techniques became had high resolution, now we know that indeed they have, neurons are individual cells, but that is because the synaptic leg is microscopic. It's in the terms of I believe it's like within the nanometer scale that this distance between neuron and neuron. So you understand why that debate at some point was had. But here you have which one? So here is a mitochondria at a synaptic bulb, and these are vesicles with neurotransmitters. Here is the synapse. Here is another neuron. Which one is the presynaptic neuron? This one or this one? This one or this one? I want to remind you, this is, yes?
Oh, the one on the top. Like,
Okay, yes. So why do you think that's a presynaptic neuron? Because it says he somewhere, right?
well, because I would assume that it hasn't kind of how I think about it is like charge. So it's not charge up there. So once it's shooted, it should go to the synaptic.
So you're saying
Kind of.
that so you're saying that the it hasn't released its neurotransmitters.
Yes.
Okay, that's a good way of determining that, because this is an axon, right? The axon is the one that sends the neurotransmitters, the signal, and this is the one that receives it. And this one receives it through receptors. So here, these vesicles fuse into the membrane, and then so now we have neurotransmitter floating. It's no longer in vesicles; it's just floating near the synaptic cleft. And at the postsynaptic membrane, these are dendrites at the postsynaptic end. There you only have receptors, kind of a receptor kind of listening to a signal. And if, it's a dopamine receptor, then it receives a dopamine signal, then it undergoes, the receptor opens a channel or something, or some reaction occurs. So that's a good way of telling. That is a good way of determining whether it's this presynaptic or postsynaptic. The neurotransmitters, yes, because the neurotransmitters, the presynaptic neuron releases neurotransmitters into the synapses. These neurotransmitters bind to receptors, and from there on, response, a signal occurs. Okay, this is classification of neurons that either based on the number of processes that come out of them. So here you have a bipolar neuron. That kind of makes sense. Two processes, 1 and 2. A unipolar neuron, one process. You see one process, this little process here, even though it looks like there's two, they're all coming from the one process. Your sensory neurons are like this. Basically, whenever you feel something, this sensation, so this is, so this you get an input here, you touch, you feel the touch, then that signal travels down this axon. It reaches the cell body, and there's some integration happening here, and then it continues to the central nervous system. It goes, so this is your skin, this is the central nervous system, and from there it sends a signal saying, hey, somebody touched my hand. But the point being is like, one is unipolar because this is the axon. There's only one process coming out of the cell. That one process divides into two immediately, but there's only one process coming out of the cell. This multipolar neuron, there's many processes. We just lost count, so we just call it multi. 1, 2, 3, 4, 5, 6 processes. That's a lot of processes. Anoxic neuron, Jesus, there's not, this one, there's not an axon, so anoxic, no axon. It has a lot of processes. However, and you will kind of learn if you study, if you go into neuroscience, you will kind of understand why they went this route. Because these are populations of neurons that usually are found within a particular cluster. And the particular cluster or the particular structure where they're found, they have the The particular region has a particular function because of the shape of the neurons. Now let me give an example. Where are we? Oh boy, oh boy. Okay. So let me give an example. For example, this neuron you can understand is good at integrating signals because it can make a signal, make synapses with several other neurons, right? And then it will sum those signals and decide only and have only a on or off output based on the thousands of signals that it integrates. This particular one is kind of like a 1-to-1 ratio. I get one signal and I send it. I get this one signal and I send one input out the other end. Bipolar neurons, they are the anoxic neurons. They are kind of surveying the environment around them and sending local signals within the environment around them. Whether these ones, you can have one region of your brain integrating a signal and then sending it to another region saying, okay, this is the signal that we got, this is what we decided. Okay. That was in terms of shape. Neurons can be functionally, they can be sensory, motor, or interneurons. Interneuron association neurons, like the little one that we saw, without any other any axon to send a long signal towards another region, but just locally, kind of integrating the signals that are occurring locally. So you can think about this. I believe computers. If you're familiar with computers, they work on nodes. You might hear this notion of a node. So the computers also, there's a lot of signals occurring at this end that they integrate at several points, and your neurons are designing in that particular way as well. So we talk about sensory neurons, we talk about motor neurons, and sensory, the afferent, motor, the efferent or exit, sending a signal outside of the central nervous system. And the interneurons, they're local. They're not going or coming anywhere, signal local that's staying there. Located in the central nervous system. And okay. So here they are. This level of detail what do we have here? The peripheral central nervous system, and these are the receptors. Receptors can be, okay, so here are these are this is the visceral portion and this is the somatic. The visceral, you can imagine, is just receptors within your body, and then they send it to the central nervous system. Interneurons integrate these signals. Then they have a motor, an output to visceral motor neurons, smooth muscle that visceral motor neurons within the central nervous system will send a signal through preganglionic fibers. Just like a presynaptic neuron, we have preganglionic fibers. So these are the fibers before the ganglia. Here are the ganglia. And the ganglia send signals through postganglionic fibers. What is a ganglia? A ganglia is a collection of neurons in the peripheral nervous system. Okay, so your nerves the nerves that allow you to do everything, usually their cell bodies, especially for the sensory and in some cases the motor, the cell bodies of those neurons are not can be found within the not outside of the central nervous system. So these cell bodies, they kind of group together hundreds of neurons. They collect and they form these little bundles of neurons called a ganglia. Probably heard the concept of ganglia. This is what a ganglia is: a collection of neurons outside the central nervous system. Here you have these motor neurons affecting a function. These motor neurons performing these visceral motor neurons performing a function, sending input to these ganglia through preganglionic fibers. The ganglia, the collection, the neurons within the ganglia, respond to that signal, and they themselves send the signal to smooth muscle, glands, cardiac muscles, adipose tissue. That sensory system that it's not under your control, the one that's under your control, we kind of talked about it as well. The only new part here is the interneurons associating those signals. There's a lot that we're gonna cover this week, so I'm going to juggernaut. I'm not gonna lose 1 minute out of this. Neuroglia of the central nervous system. What is neuroglia? It's not neuron. It's basically cells that are not neurons, right?
Yes.
Awesome. So what cells do we have that are not neurons? We have ependymal cells. We have astrocytes, oligodendrocytes, and microglia. Learn them. So let's see if we have a photo. Okay, so ependymal cells, they line up the central canal and the ventricles of the central nervous system. What are the ventricles? What's the central canal? Basically, the central nervous system has a empty, cavernous environment, empty interior kind of, where central canal kind of flows. This fluid is present and inflows. The central canal is kind of intuitive. It's in the middle of the spinal cord. But the ventricles themselves, there are like these openings, these holes, and then we have 4 of them. And we'll take a look at them in the future, where cerebrospinal fluid resides. These are lined by ependymal cells. Ependymal cells which have cilia, which create motion and move the fluid along. Ependymal cells, they do that. Okay. Now we mentioned we have. I wanna provide the function of this so that it will help you classify, because the cells of the central nervous system, the neuroglia of the central nervous system, they have analogous cells in the peripheral nervous system with different names. That way you have to study more. Okay. No, it's for accuracy. It's for accuracy. So we have the cells that myelinate, the oligodendrocytes. They myelinate, so they basically these long axons that we were talking about, where the signal, this electric signal is being sent through long distances. We mentioned an axon can be the length of your leg, because the cell body of that axon resides near the vertebra. Now, through evolution, we have devised a system that myelinates. Basically, it provides a buffer, or it's intuitive within an electric cable, where you have a kind of some rubber surrounding it to insulate it, so you don't lose the signal that's being propagated. Same goes for neurons. You have this cell that basically wraps around and myelinates the axon. That's the oligodendrocyte. An oligodendrocyte, one single oligodendrocyte can myelinate many axons. Then you have astrocytes. Astrocytes maintain the blood-brain barrier. What's the blood-brain barrier? The name is fairly descriptive. So we have blood vessels everywhere, right? Because all of our cells need oxygen. We bleed out of every corner of our body because all of our cells need oxygen to live. No oxygen, they die immediately. We also have cells, vasculature within our brains. And so we have a barrier. Basically, every blood vessel within your brain has a layer of astrocytes surrounding it. So it creates like a barrier around every single one of these blood vessels. That way, we can better regulate what goes from the blood vessel into what you know as your brain, the parenchyma of your brain. The reason why we do that is because whenever we eat something, let's say I, eat dopamine. I just take a bunch of dopamine, eat it. Or I have a meal. Whenever we're digesting, we take all of those nutrients, everything we found, and we put it on blood vessels. Those blood vessels go to your liver and then to your heart and then everywhere, right? What if this barrier wasn't present in our brains? Then everything that we ate will travel through the blood vessels in our brain, and just by just natural diffusion, it will diffuse into your brain. So immediately after you eat, you will feel like an immediate reaction. Your neurons will react. But that's not what happens. We have a barrier built by these astrocytes. Basically, their little feet, they send them out, and let's say this was a blood vessel and this was an astrocyte. So the astrocyte sends its little feet, and it just wraps the blood vessel, just like that. So now we have this barrier. And in order for substances from the blood vessel to enter the brain is everything outside of my hand. In order for that to happen, we have to purposely kind of through receptors send it from the blood vessel into your brain parenchyma. So that's very pivotal role, maintenance of the blood-brain barrier. I'm gonna take I'm gonna take extra 20, 30 minutes today because we have to cover 2 chapters this week. Astrocytes provide structural support, regulate ions, nutrients, and dissolved gases through this function, absorb and recycle neurotransmitters. That they play a big role within the synapse itself. Whenever within a synapse, you have the presynaptic neuron, you have the postsynaptic neuron, and you have an astrocyte playing a role in there. An astrocyte regulating that synapse is functioning at its best, because every time we release a neurotransmitter in the postsynaptic and in the synapse, that neurotransmitter is going to stay there if you don't do something with it. Once it performs its function, it just chill, and then whenever the receptors kind of go back to its normal environment, then they will once again activate it. The astrocyte is there to help regulate that. Sometimes it does this by reuptaking some of these neurotransmitters. The astrocyte itself kind of clears, it vacuums the synapse, taking away all of these neurotransmitters, it vacuums it, and now the synapse is ready for another round. So basically now the cell is ready and receptive to another round of neurotransmitter release. It also forms scar tissue after injury. Whenever, you can have injuries within your brain. These injuries can be dead fatal. Your body has developed a system to quickly contain, react, and clear this injury based on the fact that any injury can be fatal. So they do so, astrocytes, which are all over your brain, they can, all over your central nervous system, they can contain an injury site beyond the usual blood clotting process that we have. Astrocytes will create a barrier around an extra barrier around the injury site to prevent further damage towards the brain tissue. And this barrier will help promote recuperation. Now, we mentioned that usually, even though it might not prove fatal, usually any damage to your brain will prove usually a life-altering, because the function of your brain depends on the connections that you make, and the connections that you make will depend upon a lifetime the connections of the neuron depend upon a lifetime of experience. And that lifetime of experience cannot easily be replicated. Your healing process is just not that sophisticated to say, okay, a neuron was there, let's put one there, and that neuron was talking to those neurons, so let's put them there. That's not how the process works. The process works, once again, through you basically start anew. And those are the challenges. That is, if what part of the healing process involves scarring, scar formation, no neurons present there. So you have to build up this. It can have a sequela for like years and so on. Okay, so those are the astrocytes play these like very pivotal roles. Microglia, microglia are nothing more than macrophages, which are nothing more than monocytes, which are nothing more than white blood cells. I want to put that into perspective, because pretty soon this naming will get out of control. But I want you to know that these are white blood cells, that they just happen to be white blood cells of the central nervous system, of the brain. And they will do what white blood cells do. They go and there are foreign invaders, they go and attack, and whenever they attack, they do what white blood cells do. That's just the cells of the central nervous system. The peripheral nervous system has analogous cells. Schwann cells are the analogous cell of the oligodendrocytes. The difference between these is that a Schwann cell cannot myelinate several axons, which myelinates one. Satellite cells, they are analogous to astrocytes. They will be present. Now, we mentioned that in the peripheral nervous system, cell bodies of neurons are only within this ganglia, this collection of neurons, this round collection of the neurons. And within this ganglia, you have satellite cells. And these satellite cells, they're there performing the function that astrocytes perform in the central nervous system. Repair and just regulating the synapse, basically. We don't have we do have an analogous cell to microglia. However, we don't mention it because it's just a macrophage or a white blood cell, basically immune cell. It doesn't have a specialized name. Here is the ependymal cells lining the central canal of the central nervous system. This is the center of your spinal cord that you're looking at. Here is the view of that. This is the center of spinal cord filled with cerebrospinal fluid that contains neurotransmitters, that contains ions, that helps regulate your brain homeostasis. This is lined by ependymal cells. These ependymal cells play a role in the motion, and the specialized portion of the ependymal cells within a region called the choroid plexus, they are the ones responsible for making cerebrospinal fluid. Here you have a view of how astrocytes create the blood-brain barrier. So here you have an astrocyte. You see this blood vessel? Is completely surrounded by the processes of astrocytes, so that only whatever we want to go from the interior of the blood vessel into the brain will go, because this blood vessel is going to contain glucose. It's going to contain everything that we digested, everything that gets transported, hormones, ions, oxygen, all of those things, proteins, everything is going to be just floating, and we just want what we don't want every heartbeat, every meal to have an immediate effect within your neurons and neurotransmitters. So we completely wrap them. So we limit what makes it outside. Obviously, glucose, and that's what we want. Oxygen, that's what we want. But other things, they're highly regulated. Here's an oligodendrocyte myelinating two different axons. Here is the central canal surrounded by ependymal cells. Here are the Schwann cells, satellite cells. Let's see where we're at. 712, and we are in the middle. Let's see if we can cover. We'll cover up to the action potential, okay? So that's gonna take me until 7:30. If you have to go, understand, but I hope you revisit this. We just have a lot to cover this week, okay? But let's go to the potentials, and for this, it's a complex concept. So I hope that we can just we need your attention at the moment. Okay, so the transmembrane potential is basically the potential difference between the interior of the cell and the exterior of the cell, right? What's the potential difference between those two? That's what it is. So let's say if it was positive 100 in the positive 100 volts in the exterior of the cell, and it's positive 200 volts in the interior of the cell, then the difference is 100 volts, right? They're both positive, but the difference in voltage is 100 volts, and that's negative 100 volts in the interior of the cell. So what's going to determine these charges is going to be ions. You have the different ion concentrations in the interior of the cell and the exterior of the cell. The main ones, and each ion is going to have its own type of concentration difference. So this that we were talking about, what's the difference in sodium between the interior of the cell and the exterior of the cell? That's going to be a number, right? Usually there is more sodium in the exterior compared to the interior, and sodium is positive. That's another thing. We have to understand which are positives and which are negative. Sodium, potassium, hydrogen, positive ions, calcium, positive ions, negative ions, it's going to be chlorine. Chlorine is going to be your negative ion, okay? So all of these ions that I mentioned, they have different concentrations between the interior and exterior. There's more potassium in the interior of the cell. There's more sodium in the exterior of your cell. And you sum up all of these differences in charges, then you end up with what's the transmembrane potential? What's the potential? What's the charge difference between the interior and the exterior? It's negative 70 millivolts. That's what a cell at rest usually is at. It's at negative 70 millivolts, meaning that the interior of the cell is negative 70 millivolts compared to the exterior of the cell. It means that the exterior of the cell has 70 millivolts more than the interior of the cell, or it's like 70 volts more positive. Okay? The sodium-potassium pump plays a role within this. So here you have here you have this here you have your sodium-potassium exchange pump, and you also have these leak channels. So what do these leak channels? What's the role of these leak channels? The leak channels, we mentioned that there's going to be these differences there's going to be these differences in ionic concentrations, which are differencing not only in concentration, but they're also, since these are charged molecules, these will be differences in charge as well. So based on these differences, here we have ions that, based on this difference, here we have ions that that will flow based on these electrochemical gradients. If there are channels that are open, now these ions, they need channels to open. The lipid bilayer is impermeable to ions. Like ions can't cross it. Ions have a charge. The lipid bilayer is hydrophobic, so ions, they'll repel. There's no charge in there. So you need a channel in order for these ions to move across the bilayer. There are channels that are just readily open. One of those channels is the sodium and the potassium leak channels. What is the difference in potassium between what is the difference in once you sum up all of these events that are occurring, the natural leakage of sodium, of potassium going to the exterior, we mentioned, because potassium, there's more concentration of it. So once you open that channel, it would just naturally flow to the outside, right? Potassium won't go inside because there's already a lot of potassium inside. The opposite is true for sodium. There's a lot of sodium outside compared to the inside, so sodium will flow in this direction. Now, this once you add up all, and the sodium-potassium pump, in turn, is kind of having the opposite effect. It's creating these gradients that were originally there. So this difference, the fact that there's more sodium outside and there's more potassium inside, doesn't just happen by accident. And the fact that it's always that is kind of generally the case, it happens because we're constantly generating this gradient. We're constantly pumping potassium to the interior of the cell, and we're constantly pumping potassium out of the cell. This is constantly happening. This is constantly happening by this pump that constantly uses ATP. This ATP that if we were to just like stop the production of ATP, then the sodium-potassium pump would no longer be able to create this gradient. So the fact that there was more sodium inside and potassium outside, that would no longer be the case. So once these gradients the gradient is no longer there, then basically it's like a cell phone without a charge. There's no charge to perform a function. And in this case, there's no charge to perform that function. So that is why we are constantly needing oxygen, because ATP, in order for the process of ATP formation, requires oxygen, and ATP is constantly needed to create this gradient, to basically charge your cells. If they weren't there, then the charge goes away. We can no longer perform a function. Basically, we can no longer fire an action potential. You don't fire an action potential, all of your basic life processes deteriorate. But anyway, we create these gradients and we have these leak channels. We create these gradients, and these ions are naturally leaking inside on the outside. And the sum of all of these different charges and different concentrations is what leads to the membrane potential. If we look at the electrochemical gradients individually, so we look at the electrochemical gradient for potassium, or we look at the electrochemical gradient for sodium. what happens if, so this is the electrochemical gradient of the cell naturally. We mentioned there's more potassium in the interior of the cell. Potassium, because there's a lot of potassium in the interior of the cell, it will naturally try to go to the exterior. That's a gradient, that's a potassium, that's a concentration gradient. And the electrical gradient, potassium will naturally want to go into the interior of the cell because the cell is, after all, negative 70 millivolts. So overall, once you add up all of these forces, the concentration gradient, the electrical gradient, you end up with the electrochemical net gradient. And the electrochemical net gradient of potassium is that it generally does want to leave the cell. And whenever it does, whenever you allow potassium to leave the cell, it will take away with it. So this was a negative 70. It will take away with it 20 millivolts of charge, right? All of the positive signal of potassium that it leaves, it will leave the cell at negative 90 millivolts, and that's the, it's called the electrochemical potential of potassium. So if you basically, if you open all of the potassium channels within the cell, what would potassium do? It will leave until the cell is negative 90. It will take all of its positive charge until the cell is negative 90 millivolts. And at that point, it is at equilibrium. At that point, the concentration gradient and the electrochemical gradient cannot reach a balance. So there's no longer any movement of potassium, right? Because now it's at a happy place. The happy place for potassium is negative 90 millivolts. Why is it negative 90 millivolts? Is because it's the forces that are determining whether potassium moves in and out. They are forces of concentration and forces also of charge. And the force of concentration is saying, potassium, get out of the cell, there's too much of you inside. And the forces of charge are telling, hey, positive potassium, stay on the cell is negative, we need you. Basically, potassium can't go outside, it's too positive. It's like positive going towards the positive, it's not against the gradient. Let's do the similar kind of experiment with sodium. Here you have sodium, its chemical gradient and its electrochemical gradient. The chemical gradient was, we mentioned the cell naturally is negative 70 millivolts. You open these arrows, I hope you start kind of intuitively sensing that these arrows means we have open channels, because ions can readily cross the membrane unless specific channels are open, and then they can travel through those channels. So we open channels, and now the chemical gradient, there's more sodium in the interior of the cell, in the exterior of the cell. So yeah, it will gonna go inside. And the electrical gradient is also favoring sodium to go into the inside, because there is the interior of the cell is negative and sodium is positive. Sodium and potassium are positive. So basically this positive charge wants to go into the interior, and based on its chemical and its electrical gradient. You sum up all of this, and then whenever you open channel, sodium just rushes into the cell. It will rush until the cell is positive 66 millivolts compared to the exterior environment. That's whenever you will reach this balance of concentration and chemical gradient. So what I want you to understand out of here is what happens when I open sodium channels or when I open potassium channels. Whenever I open potassium channels, potassium is gonna go out of the cell. Whenever I open potassium channels, positively charged potassium is going to take its positive charge outside of the cell, leaving the cell a little more negative. It used to be negative 70, now it's negative 90. What happens if I open sodium channels? When I open sodium channels, sodium is gonna rush into the cell following its electrical and chemical gradient. What happens when I open sodium channels? The positively charged sodium goes into the cell, and that positive charge and it will go into the cell until the cell is cell is positive 66 millivolts compared to the stereo of the cell. That's whenever it reaches equilibrium. Membrane contains, all right, let's leave it at that. We're gonna look at the understanding this puts us in a great position to start understanding action potentials, okay? Well, thank you for sticking around. This is the nervous system. The nervous system is challenging, but it's interesting. It's good to know why you are the way that you are. Goodbye, everybody. Oh, any questions? Nothing? Awesome.